So then you're a PhD candidate with the Department of Pathology.
Yes, I am.
At Dow.
Yeah.
It also says that you do research in the field of pancreatic cancer at the Beatrice Hunter Cancer Research Institute.
Yes. Yeah.
What's that? Something I've heard of.
So it's one of the institutes that actually provides funding for graduate students at Dalhousie, or actually in the Maritimes. So they get money from CIHR, so the Canadian Institute for Health and Research,
Yes.
from Terry Fox, from the Canadian Cancer Society, from some Dalhousie. So they kind of combine all the money and they put it in a single pool, and then they redistribute that to graduate students. So they actually funded my project as well, along with the Pancreatic Cancer Society. So that was kind of the idea when you guys collaborated with the institute. And I was fortunate that I was working on pancreatic cancer, and yeah, so that's basically what the institute does.
Could you give us just a little bit of an overview of your background?
Myself, so I did my undergraduate studies at Dalhousie with an honors degree in microbiology and immunology, and then I joined the master's program in the Department of Pathology. And now I'm doing a PhD, so kind of the next step of a master's in the Department of Pathology, and I've been working with Dr. David Wiseman. So he's a professor in the Department of Pathology as well as the Department of Biochemistry and Molecular Biology at Dalhousie. So that's me in a nutshell. I was born in Lebanon and raised in Lebanon. I moved to Canada at the age of 17. So, as mentioned, my name is Moamen. I'm a PhD candidate with the Department of Pathology. Our lab focuses on studying pancreatic cancer and trying to identify new targets that we can use against pancreatic cancer using molecular and genetic approaches. So my talk today might be slightly different from what you've seen before. We do research, so we take different approaches to pancreatic cancer than, let's say, doctors or others. So before I go on, I just want to give you a quick overview. Basically, through this talk, I'll try to give you an idea of how we can define new targets for pancreatic cancer, how we can find ways to predict treatment responses, how we can stratify patients upon diagnosis, and also how feasible it is to translate what we do on the bench, so basically what I do, to the bedside. That is, transferring what we do on the bench to a drug that we can give to a patient. And again, we'll be using two different approaches, a molecular approach and a genetic approach. I'll be discussing what this means soon. Now, before I go on, just a couple of announcements. I'm not a physician, so I'm not the guy that writes prescriptions, and I'm the guy that does the experiments to come up with a new medication. And also, please, if you have any questions, please stop me. Sometimes I get too excited, and throw in some scientific jargon that you guys may not need. Okay, so I'll start off with this slide. This slide is showing on the left-hand side here a child who has a big tumor on his back. His name is Jesus Rodriguez. He was born, in the picture, he weighed only 12 pounds. His tumor weighed 15 pounds. On this side, we have Steve Jobs. He had pancreatic cancer. His tumor was not really that big. I mean, we're talking about maybe walnut-sized, probably smaller. Steve Jobs died, unfortunately. Jesus did not. In fact, he is fine, alive and well today, as you can see here. Now, the message I want to convey on this slide is basically it does not matter how big the tumor is. It matters if it's invasive or not. Okay, so the cancer here is non-invasive, so it did not matter how big the tumor was. Compare that to pancreatic cancer, for example, which tend to be invasive. And so, as you guys know, that pancreatic cancer patients have one of the most or the worst patient mortality. So if I give you an example, if 100 individuals were diagnosed with pancreatic cancer, maybe 5 years down the line, about 4 individuals will be alive. So that represents a 5-year survival rate of 4%. Yeah, and so fortunately, pancreatic cancer is not very common. In Canada, there's about 5,000 cases a year, every year, plus or minus, and that's why it only accounts for, or stands at the 4th leading cause of death in North America. As mentioned, the 5-year survival rate is 4%, and the pancreatic ductal adenocarcinoma, so it's known as PDAC, is actually the most common form, and it tends to be aggressive and metastatic, especially at the time of diagnosis, which tend to be late, at which point the disease has metastasized or at its advanced stages. The adjuvant, so surgery is available, of course, and usually patients are given chemotherapy after the surgery, and that increases the survival rate to 25%, but we're still at the low end in terms of survival rate. You can compare this to breast cancer, for example, which is 88%, so you can see the massive difference in survival. And now the current treatments that we have are kind of a bit limited, and they have variable efficacies. So they work on some patients, they don't work on others. Now, when I say metastatic or invasive cancer, what does that mean? For us, because we're researchers, we tend to zoom in into things. So we like to look things at the molecular levels, at the cell level. And the way we look at it is as this. Basically, a cancer cell is actually present within the duct of the pancreas. So if you imagine the pancreas is made out of ducts, and the cancer will start in one of these ducts. And so the cell will start growing and growing and growing. And as some of you may know, cancer is the disease of the DNA, or the disease of the genome. So at some point, the cancer or the cancer cells will make changes to the DNA, which allow them to become invasive. And that's shown here. So these cells are now invasive. So now they can make their way through the obstacles, they can carve their way and make it all the way to the circulation, to the blood, and then they can metastasize to other organs. In the case of pancreatic cancer, mostly it's the liver, the spleen, the bone, or sometimes the lungs. And that leads to patient mortality. Now the idea is, now the ability of the cell to do this, to invade, to get to the blood, undermines the difference between a benign cancer versus a malignant cancer. And so if you go back to the slide I showed you at the beginning with Steve Jobs and the child, remember non-invasive versus invasive? So that's exactly the same. Benign tumor means non-invasive. Invasive cancer means it can be malignant and it can metastasize. So what we do is basically we try to target key elements that are required for that invasion process to happen. And those would be potential targets for therapy. Now, just to give you an idea of when I say an invasive cancer cell, what does it look like? This is a cell in culture, cancer cell, and I'll just play the video. And basically you can see it's moving. Now this is very fast. Now I know it may not look very fast, but for a cell that's very fast. You can see it has these spiky things on it. Now a normal cell does not look like that. It has a really irregular shape and it does not move. If it does, it'll die. A cancer cell does not die. And that's kind of the trick that cancer cells have. Okay, I'm just going to stop that. Now,
so
yeah.
Is it moving like an amoeba or something?
Kind of, yeah. Yeah, it's a special kind of movement we call the mesenchymal movement. So that's what cancer cells do. They are able to move. Usually, if a normal cell is moving, it's going to die. There's something in the cell that tells it you have to die. You cannot move, right? But because the cancer cell had changes in its DNA, then there are no breaks. So when it moves, there's no signals in the cell telling it to die, so it just keeps moving. So that's one of the characteristics of cancer, is that when they move, they don't die. The mesenchymal movement actually involves the breakage of these bonds between cells. So cancer cells are able to do that. So they can break off from each other, and then they can move to the blood and metastasize. Normal cells cannot. If they do that, the cell will immediately die. There's signals in the cell that will tell it you should die, you cannot move. Cancer cells don't have these breaks, or they have these signals in the cell. Okay? Does that make sense? Now, just switching gears a bit slightly, just going back to what I mentioned a few slides back in terms of the limitations of the therapies that we have currently. I think there are three major problems with these therapies that we have for pancreatic cancer today. Number one is toxicity. So as you guys may know, many of these drugs are really toxic. So they have a lot of side effects. And mainly because they do target tumor cells, but at the same time, they also target normal cells. And that's where the toxicity or the symptoms of the treatment come from. The other problem actually relates to the drug discovery and development process. So it's a really lengthy process. It takes about 10 to 15 years to develop a drug, from having a drug that you think might potentially be a cancer therapy all the way to a drug that might be used or given to patients. It takes about 10 to 15 years. And the other thing is the inefficiency or the success rate of these things. This kind of shows you an example. If you have 5,000 to 10,000 compounds that you think might have potential therapeutic effect for cancer or pancreatic cancer, only one of these compounds will be approved by the FDA. So the FDA would be the Food and Drug Administration. Those are the ones that say yes or no for any potential drug. So you can see the inefficiency in developing drugs against cancers, including pancreatic cancer. Now, for us, how do we look at pancreatic cancer? We look at it as three different steps. And so the first step would be, as I mentioned, cancer is the disease of the genome, disease of the DNA. So any changes in the DNA sequence means that cancer might occur. This is known as a mutation here. So this change in the DNA sequence will give rise to changes in the behavior of the cells. So remember I showed you the cell and how it's moving? So that's a change in the behavior of the cell, which involves changes in molecular pathways. And I'd like you guys to keep this in mind, because we'll come back to it later. Now, the changes in the behavior of the cell will eventually cause the cell to acquire new skills. So in the case of pancreatic cancer, it'll make the cells more invasive and more metastatic. Now, the issue is that the transition between these steps are not always well understood. And if we want to develop therapies against pancreatic cancer, we have to define these molecular pathways, because those are the ones that are specific for the cancer and are not probably present in normal cells. And that way we can develop what we call a target-specific drugs. That's something that we don't have right now. The therapies that we have right now they go after tumor cells, but they also go after normal cells. And so that's the idea. Now, molecular pathways. It sounds easy, but cancer probably has the most complex molecular pathways among all diseases. This is a simplified image, very simplified.
I'm so glad you simplified it.
Well, I will simplify this further. But yeah, it's a really complex disease. But I will simplify this to this, and hopefully that will be a bit simpler. Basically, molecular pathways are broken into two different components. You have a regulating component, so this part, and you have an executing or executioner component. The regulatory component is the one that gives orders. So do this, do that, don't do this, don't do that. The executioner is the one that does the job. Now, in pancreatic cancers, one of the major regulators in pancreatic cancer is there's two of them. There's a bunch of them, but two of them are KRAS and TGF-beta. So those are two regulators in pancreatic cancer that are highly prevalent. Now the problem with, well, you can say, okay, let's target these guys and see maybe those are a good target. The problem with these is, with targeting these elements, is that those, although they are involved in cancer, however, they are also involved in normal cell function. And so we also go back to the same toxicity problem. The better option is to go after the executioners, because those tend to be more specific for the cancer. So our lab works on a specific one of these executioners called P11. And this executioner tend to help cancer cells become more invasive. So it kind of, for us, signifies the potential target that we can go after in pancreatic cancer. And our lab has actually worked quite a bit on this executioner, P11, and had shown that it's involved in leukemias, in some skin cancer or soft tissue cancers, in lung cancers and colon cancer. However, we have not looked at pancreatic cancer, so that's something we have not looked at. And when I joined the lab, that was the idea, that I could look at pancreatic cancer for my PhD. Now, what do we know about this executioner, P11? I call them the no and the no-nos. We know that it's important in many cancers to make them invasive and metastatic, but we don't know if it's involved in pancreatic cancer. And what we also don't know is what molecular pathways regulate this P11, this executioner. And so one of the reasons why we wanted to study P11 was actually a study published in 2009 here, showing that the expression of P11 was high in pancreatic tumors compared to normal pancreas tissue. And this expression was increasing as the cancers became more invasive. So as you go from the left to the right, the cancers become more invasive, and you can see more brown color, which means there's more of the element, more of P11, which suggests that it might be posing an advantage to the pancreatic cancer. So that's why we wanted to take this further. What I do right now is I study pancreatic cancer using 3 different approaches. The first approach is using animal models. So we use mice that have pancreatic cancer, and we use them to study human pancreatic cancer. We also use cell culture studies, so that's in petri dishes, and we also use we're hoping to use some human samples, pancreatic cancer samples, to validate our hypotheses. So we'll just start off with the animal models. So see what I've seen so far with the animal models. So this is the animal model that I have. Don't worry about what it says on the slide. It's quite complicated, but basically these mice are designed or genetically designed to develop pancreatic tumors. The tumors that these mice will develop closely mimic the human pancreatic cancer, and the reason why is because they were genetically modified to simulate or to resemble the human pancreatic cancer. And so these mice will develop tumors, and we can isolate these tumors and study them. And so that's exactly what we've been doing. We're fortunate enough to get this model from Harvard University, and it's actually one of the best clinically relevant models to study human pancreatic cancer. So I was really fortunate. We are really fortunate for that. So what we did, basically, I isolated these pancreatic tumors from the mice, and I looked at the expression at this P11, this guy here. And I looked at normal pancreas and also at the tumor. And what I found is that there's more of the P11 in the tumors versus in the normal tissue. So the darker the band, the more there is of the protein or the element. So you can see that these two have way more than these two, telling us that there is more P11 in the tumors. And also on this image, there's more of the dark pink or purplish color on this slide compared to that slide. So that's telling us that there's more of P11 in the tumors, and it's telling us that it might be advantageous for the tumor and potentially can be a target for us to go after. Yes, it's the same. So now we're actually in the process of deleting P11. So what we can do is we can take these mice and we can delete the gene responsible for P11. So we'll take it out, and then we can look at how that affects pancreatic cancer in these mice. Okay, and so the way we look at how the loss of that gene of P11 affects the growth of the pancreas, of the pancreatic tumor. We can also look at how it affects how invasive the cancer is and how metastatic it is. So we're actually in the process of doing that, but we're hoping to see what we saw with lung cancer. So this is what we saw with lung cancer. I'm just going to use this as an example, and we're hopefully expecting to see more of the same. And so if you look at this graph, this line represents the tumors that did not have, that were deleted off P11. So there was no P11, nothing. So the tumors did not grow. So just straight line. However, if the tumors had the P11, the tumors grow quite fast. And it's basically shown in these images. So at the bottom you see the tumors that did not have P11. So you delete the gene, there's no P11, and you see the tumors are really tiny compared to the tumors that had the gene, and so the tumors grow quite large. So that's very promising. Again, we're in the process of doing that, and we're hopefully expecting to see the same of the same thing. That's kind of summarizes what we're doing with the animal models in terms of the cell culture studies. So what we do in petri dishes. So this image kind of shows you what we do in a nutshell. We culture human pancreatic cancer cells in petri dishes like this, and we grow them, and we can do experiments on them. And so one of the things that I did was I tried to delete the P11 in a pancreatic cancer cell, as shown here. So the absence of the band basically signifies that it was deleted or was depleted. And what we saw was, when we did that, there's about a 60% reduction in how invasive these cells are. So you can see that this protein or this element or this executioner is contributing or is important for the invasiveness of these cells. And that's why the loss of it is causing a reduction in the invasion of these cells. So going back to this, now we know that it is involved in pancreatic cancer. We've done more stuff, but it's kind of in a nutshell. What we don't know is that what molecular pathways regulated. And without going into details, basically what we found is that P11 is regulated by the KRAS pathway, which I mentioned earlier on. Again, showing by how dark the band is. So see there's increase in the darkness of the band, meaning that there's more of it that's being regulated by the RAS path or the KRAS pathway. Also regulated by the TGF-beta pathway. Again, it's one of the pathways involved in pancreatic cancer. And we're also able to identify another executioner along with P11 called HMGB1. So this is still in the works, but basically it's a really potential target as well for therapy. HMGB1. And now in terms of the third approach, which is still in the early stages, but basically what we'll be doing is we'll be gonna be collaborating with Dr. Wei Wang and actually Dr. Molinari in terms of looking at tumors isolated from patients and look at P11 in these patients and see how that relates to their survival, to their outcome, to their basically to how invasive their cancer was. And the reason for this is because we want to validate what we're seeing with the mice and in the culture in humans, because that kind of explains or validates the hypothesis that we're working on. Yeah. Again, so why is this important? It's important because these executioners, being P11 or HMGB1, are potential targets for therapy. And also, they can also be used as biomarkers. So by biomarkers, I mean something used to predict outcome. So for example, if a patient comes in, we can look at the P11 and see, okay, low levels, that's a better outcome. We should expect a better outcome versus high levels, meaning a worse outcome. Then we can deal with it in a different way than a patient with a better outcome. Yeah, this is the model we're working on. I don't want you to worry about it. Basically, I just simplified it in here, showing that our two executioners, P11 and HMG1, are being regulated by two different pathways. Both are implicated in pancreatic cancer. So to conclude, as I said, it's regulated by these two pathways, which are both implicated in pancreatic cancers, and that we have identified these executioners that are potential targets for therapy, P11 and HMGB1, and they can also be used as prognostic markers or biomarkers to predict the outcome of patients. So at this point, that's what I've been doing in a nutshell. I have more slides to kind of discuss what else is going on in pancreatic cancer research. But if you have any questions so far, please do ask.
Has anybody looked at targeting blood as a treatment? So this cancer cell is detaching and then traveling instead of
So targeting the cells in the blood, is that what you're saying?
Targeting blood against the cells.
So you mean like go after the cancer cells in the blood, or you mean just the blood itself?
Use the blood as
Oh, okay, so you can use immune cells. So immune cells are present in the blood, and you can use these against the cancer. There's a few labs that work on this. We don't actually work on it, but it has proven, it has shown some success in terms of using your immune system to target the cancer as well. But yeah, that's yeah.
Did you say that you removed the P11 in your experiments?
Yeah.
You mean you went back and re-engineered the mouse? Or the tumor was growing with the P11 and you changed the existing tumor?
So that's a good question. Great. Yeah, so the way we do it is actually we have another mouse which P11 is actually deleted. So there's no P11. And so we have to actually cross mice together to get, which is a really lengthy process, by the way. Takes about 4 or 5 months at least. But basically, at the end of the day, we'll get a mouse that will develop pancreatic cancer. So it has all the gadgets that I showed you in my model, but also does not have P11, right? And so that's our way or our approach of seeing how the loss of P11 affects the growth of the pancreatic tumor. Does that make sense?
So the P11 has no function in normal cells?
It does have physiological functions. So it does have functions in the blood, physiological functions in terms of removing the clots, so in terms of dissolving the clots. However, its function is relatively redundant in that system. So if we do decide to target it, I don't think that'll be an issue in terms of affecting the clots or how clots form in the blood.
What could possible therapies be? What would some examples of possible therapies be if this turns out to be correct?
Well, if that turns out to be correct, and we can take it further, there's multiple approaches you can use for therapy. So you can use something we call antibodies. So those are molecules that actually will go after your element and bind to it and stop its function, basically. You can use something we call small molecules. So those are smaller than antibodies, small molecules. They can also bind to your molecule and also inhibit its function. So those are the most common approaches that are used. You cannot genetically delete it from cancer. This is not possible. At this point, it's not. In 10 years, it might be, but at this point, it's not. Yeah. Does that answer your question?
So
Yeah.
he's targeting the P11. You said the P11 was in other cancers initially.
Yeah.
Has any work been done on those other cancers in regard to targeting it? And if so, what's the outcome then?
So it's actually being done as we speak. Because our lab works on that P11, but also works on different cancers.
Yes.
So I'm the person who's working on pancreatic cancer, but there's other people in the lab working on lung cancer and on breast cancer as well, and colon cancer as well. So we're still in the early stages. We haven't actually got to the step where we're actually targeting the P11. At this point, we can only genetically delete it. So like I showed you, just remove the gene. But in terms of targeting, we haven't got to that step yet. But these things have to be done beforehand, before proceeding to the next step, just basically for validation purposes, because you cannot just go into the next step. It can be really expensive in terms of funding, so you have to make sure that you cover all the bases. Okay, we have this model, it works fine. Okay, we have this, let's go to the next step. That's usually how it works. It's very difficult to jump steps. You can't make the process faster, unfortunately. You have to finish a few things, say, okay, we're done this, let's move to the next step. That's the way it works in terms of funding and where the money comes from, because that's the only way we can keep going. Yeah, does that make sense?
You basically you have two lineage of mice.
Yeah.
And you're making hybrids.
Exactly.
Okay. And they are pure breed.
Yeah.
Okay. What's the price of one mouse?
The price?
Yeah.
Well, so we
I guess it's patented. They are patented. They are made by Harvard. They are sold. They are not given.
No, actually, so it's something that we have in science, is that if you have a reagent, you actually are meant to share with everyone. So if I have something and there's a lab across Canada in BC that wants to use that reagent, I should provide it. That's kind of the rule we use in science. You should not charge money for it. Now, some people do, but that's so. I got the mouse for free, and basically I just bred the mouse, made more of it, right? That's the way we do it. That's not where the money actually, where the funding goes. It's for maintenance of these mice. So we have a facility that takes care of the mice. So that's where the money actually, a lot of the money goes. It goes into reagents, so stuff that we give to the mice, the stuff we do the analysis with. That's where the money goes mostly, but not the actual price of the mice. They were free. Yeah. Ideally, as I said, it takes between 10 to 15 years for any compound to make it to the clinic. So yeah, I mean, 10 years is reasonable. So, what else is going on in pancreatic cancer research? So, there's a couple things I'm going to address. The first one would be that we are trying to, or other people are trying to make changes to existing drug regimens. So the existing drugs that we have, we're trying to change them and try maybe different combinations. Thank you very much. Yeah, and so yeah, we're gonna just talk about this briefly. You guys know what clinical trials are, right? Basically, so just studies done on humans. And so one of the common drugs for pancreatic cancer or two of them are 5-FU, so that's or this is also known as edurocil, and gemcitabine. So those are very common. Now, there have been a lot of clinical trials trying to try different combinations of drugs. So, for example, I don't want to go into details, but basically, we're trying to go to after a combination approach. So, try different drugs, different combinations, see how that affects the patient outcome. To go to the second topic, which is I'm going to focus more on, basically trying to identify new approaches. So like what we're doing in the lab, trying to do new approaches for treatment, new targets for pancreatic cancer treatment, or even for diagnosis and then prognosis. So we'll go after that. And I'm going to introduce a topic or a concept known as pancreatic cancer heterogeneity. Does anyone know what heterogeneity is? So basically, I'm just going to talk about it anyway. So there are different types of heterogeneity, right? There are 4 different types. And when I say heterogeneity, it means differences, okay, differences. Now, these differences could be between patients. So the tumor in one patient is very different from the tumor in another patient. That's what we call interpatient heterogeneity. There's these differences among patients, and that accounts a lot for the response of these patients to the same treatment, because you'll see sometimes that 2 patients will get the same treatment, but they respond completely differently, and that comes back to the idea that their tumors are different, right? If the pancreatic cancer, someone has pancreatic cancer, it doesn't mean that your cancer looks exactly the same as the other pancreatic cancer patient. They're probably a lot different. The other thing, which is still a new concept, is that within that patient, that tumor, there's actually multiple populations. So there's a lot of difference within that tumor. So that's known as intra. So within the tumor, there's a lot of differences. So you'll see some cells which look the same, and there's a bunch of cells that look different from the original ones. There's also something called spatial heterogeneity. So spatial meaning differences in place. So, for example, a primary tumor in the pancreas is different from that, from the cells that metastasize to the liver, for example. So they look different. And also temporal heterogeneity, which is actually very important. So looking at resected tumors, so tumors that were taken out of patients, versus a recurring tumor, is the tumor that came back. There's a lot of differences in these tumors. It's actually, we call it the bottleneck effect, where after you remove the first tumor, the resected tumor, there's probably some residual cells that are left over. And so those cells will actually come back and repopulate the tumor, the new tumor. So those are completely... Pardon?
But
yeah, so they're actually different from the primary tumor. And actually, I can show you an example of that. There was a recent study published in Science, in a journal of Science. Basically, they sequenced the genome of pancreatic cancer patients. So this is a technique that 15 years ago, it would cost $3 billion to sequence a genome. So by sequencing, I mean knowing the exact sequence of the DNA of each patient. So every single there's knowing all the sequence. Okay, so that's sequencing. 15 years ago, as I said, it would have cost $3 billion. Today, it's about $1,000. So you can see the reduction in price and the feasibility of using this technique on today. Basically using that technique, what they found is that across these patients, they found some common changes in the DNA that were found across multiple patients. And they narrowed that down to 63 changes in the DNA that were very common across patients. And those 63 affected 12 molecular pathways. And so and one of these molecular pathways were the ones that we are studying, so the TGF-beta pathway and the KRAS pathway. So if we go back to our three-step list, basically what we're seeing is that change in the DNA, so there's 63 changes, those are translated into changes in 12 molecular pathways, and those are contributing to having a metastatic pancreatic cancer. And so if we can design drugs that can go after these specific 12 molecular pathways, this would be more target specific. Going back to your point about why the tumors are different, with the technology we have today, we are actually able to track the progression of a cancer cell from when it starts in the pancreas and what happens to it as it leaves the pancreas and goes to other sites, let's say the liver or the lungs. So for example, this was done for a patient, and so you could imagine this is where the cell started, and then the tumor was growing, and it got to a point where it starts metastasizing, and so that's where it actually went to the liver, and then it also went to the lung. But you can see that the cells that made it to the liver start to diverge and go to other tissues as well. And so this cell, for example, that's here is dramatically different from the germline which you started with. So that's what I mean. But now we have the technology to do that. 4 years ago or 5 years ago, this was not possible. Now we can actually track where the cell starts and where it ends up. So if we can do that for every single patient, I know it's at this point it's not feasible because the cost. But I'll go into this later at the end. But basically, at some point, this will become feasible to a lot of patients. Another study also looked at trying to distinguish patients into 3 different types or subtypes listed here. Don't worry about the names, but basically those are based on changes in the expression of the genes of these patients. Now, so what I'm trying to, this is kind of what I propose when it comes to patient stratification. So for example, if patient A comes in, we will be able to say, okay, well, this patient belongs to group 2 or subgroup 2, the quasi-mesenchymal. And so this patient then will examine their molecular pathways. As I said, there are 12 molecular pathways. So we'll say, okay, molecular pathway 1, 5. This is just an example, but just with the approach that we're going after. So let's say pathways 1, 5, and 11. So now we know what targets we can go after. And we can also stratify these patients and put them into groups. So we can say, okay, this group is high risk, and so they will require surgery, and they probably will require some kind of therapy. And if we have defined what pathways are overactive in these patients, that we can design targets that target these pathways that are specific for these pathways, let's for 1, 5, and 11, and avoid using the generic therapies that we use today. And that's kind of might avoid the problem of using the same drug, for example, in 2 different patients and having different response, because these 2 patients have different pathways active. So some of them will respond, some of them won't. And so by trying to design therapies that target specific molecular pathways that are specific for that patient, that way we can make sure that that patient will respond. So another example would be if that patient was exocrine-like, so those patients tend to have a good outcome. So we can say, okay, well, maybe these pathways are active. Maybe we can say it's a low-risk patient, so there's no need, maybe the patient will be okay with surgery only. Again, because these patients tend to have a good prognosis, a good outcome. And you can also be in an intermediate risk group where you can decide to use or not use the chemotherapy, not the chemotherapy, but the pathway-specific therapy. Now, the hurdles to these approaches, usually it's money, as you may know. So it requires multiple clinical trials because these have to be validated. It has to be studied through clinical trials, and clinical trials are really expensive. And if considered at this point, healthcare systems cannot afford it. They're really expensive. Having said that, the cost of many of these techniques is going down dramatically. As I said, sequencing pricing has dropped quite significantly in a short amount of time, from $3 billion to $1,000 or a couple $1,000. Same with other techniques, but we're not at that point where we can incorporate these technologies into our daily healthcare systems. So just going back to the beginning, can we translate the bench science that we do into the clinic? In theory, we can. It just, again, we have some hurdles that we have to, or obstacles that we have to eliminate or deal with. Mainly was basically the cost for the healthcare system. And so, yeah, to summarize, I've shown you that through our work we identified some promising targets for therapy, and I've also shown you that individual tumors actually vary among patients, and that this variability might account why we see differences in treatment or response to treatments, and may also give us an insight to how to predict outcome in some of the newly diagnosed patients. And finally, just like to thank all the people that contributed to this, and thank you for listening, and I'm happy to answer any more questions.
What kind of studies will you be doing with the human tissues?
So what we'll be doing is we'll have access to, I think, over 110 tumors resected from patients from Nova Scotia, and we'll basically be looking for P11 in all of these patients, and then looking at how the expression of P11 correlates with how that patient did. So for the outcome of that patient. So for example, if we see that P11 levels were high and that patient did not do well, versus P11 levels being low and that patient did okay or better, then there's a correlation between survival and the expression of P11. So that kind of validates what we're doing, and kind of makes things a bit faster for us to go to the next step. Does that answer your question?
Mm-hmm. So radiation, chemotherapy, does that have any impact on P11?
We don't know. I don't know. That's something that we never looked at. Yeah, that's a good question. I don't know.
Because some of these tissues will have been exposed to radiation.
Yes, yeah, exactly. So that's something we actually have to control for. So we will just look at whether that patient was exposed to radiation or chemotherapy, probably. So we can also show if P11 predicts response to treatment, right? So if high P11 would predict that patient will respond well for radiation therapy or for chemotherapy versus not responding. So there's a bunch of criteria that we'll be looking at. So survival, response to treatment, invasiveness, so stage of the disease, these kind of things. So we can actually plot that on a graph and see if there's any correlation. And again, that will help us to go to the next step where we can actually try to target P11 and see what we get.
That was an excellent presentation.
Well, thank you.
I found it very, very interesting.